Good evening. My name is Howard Edelman. I'm your host for Israel and the Jewish World. IMRC scientist Yehudit Bergman explains how the body keeps foreign elements at bay. Why can't politicians? Israeli prime ministers regularly renege on previous stances under pressure. Perhaps Yossi has an answer. Stay with us. My guest is Moshe Ronen. Welcome to the show. You've been very active in the Jewish community, and you've been participating, I think, ever since you were a student at a young age. Having a show on Israel and the Jewish community is new. It's the last 10, 12 years. What experience do you think happened that changed that, and what effect has it had on the Jewish community?
Well, I think it's sort of the new age where, media is so prominent in forming people's ideas and communicating a message. And I think it just makes things easier to, for example, tell about Israel and talk about the Jewish people through media. And you can just do it so more effectively than the intense and hard work that we had to do before, I think.
Well, you were active in what campaigns?
Soviet Jewry, Ethiopian
That's what
Jewry.
I remember you from first.
With you on the Boat People a long
right.
time ago. Quite a long time ago. But I think over the years, it's become clear that one of the most important challenges that we face is really the image, the branding, the perception that we want others to have of us as a Jewish people, of Israel. And I think the best way to do that is through spreading information in important media outlets, media venues like your show.
But, it can begin to sound like you're in advertising, like we're in advertising, and we're not. We don't try to sell a product or anything like that.
No, I don't think it's advertising. I think it's communication. I think that it's the best way of delivering in a short period of time important information, which I think at least prompts people's interests. Perhaps to look more into the issue, to study more about the subject matter, and to at least create in their minds a more definitive and correct picture of what is really going on.
Briefly, what do you think the effect is on the Jewish community itself?
Of media?
Yeah, of having this show, really.
I think it's a phenomenal effect. I think it's an opportunity to, first of all, take pride in the beauty of our achievements in the community, the achievements of Israel, to be able to focus on areas where some of us may have not had the privilege of learning about or being informed about. It's all good.
With that endorsement, thank you very much, Moshe. Really appreciate it.
Thank you for having me.
This is one of our series of interviews with innovative scientists in Israel, and particularly in the biomedical field, and particularly in connection with the Institute for Medical Research, Israel, Canada. Yehudit Bergman, welcome to the show.
Thank you.
Yehudit, you're outstanding as a scientist. I know scientists tend to be modest, but you've won prizes, etc. But what makes your portfolio interesting is you cover such a wide area. How do you do it? I mean, we'll get into some of those areas, but most people are specialists in one thing. I think you cover 4 different big areas. Who knows what else you do?
I work hard, and it's very interesting for me. So this is the drive, I think, that I like to try to see different fields and also try to understand what's similar and what's different, and then contribute to each field from thinking in the other field.
There's a connection between them.
Right.
Okay, we'll ask you what the connection is. Let's try one of them, because one of your early fields is immunology, right? Describe your work in immunology.
So in immunology, we would like to understand how the body makes the repertoire of antibodies. How do we make so many antibodies to cover everything we need in our trial to resist all pathogens, for instance? And there is a very specific process that occurs during formation of the genes that will encode the antibodies. And it's very, very unique only to this system. And there are two things that are very, very unique to it. One, that rearrangement of pieces of DNA need to take place before expression. So usually most of the genes, they are expressed from the gene itself, the way it is present in the germline, in all cells. But with the immunoglobulin genes, they have to undergo some type of a rearrangement in the genome itself, only in cells, in lymphocytes, which express and secrete the antibodies. So this is very unique. At one point, people thought that maybe in the brain there is something similar to this particular rearrangement process, but it's only unique for hematopoietic cells.
I see, just for antigens.
Yeah, so this is one unique feature of this process. The other one is in every cell, you have two genes. For every gene, you have two genes on two chromosomes, the pair of identical chromosomes. And in this particular system, only one allele, what we call an allele, one chromosome is chosen for rearrangement and for expression at the end.
So one keeps the constant and one's the variable?
No,
no?
They both have the constant and the variable.
Oh,
but
I see.
Only the rearrangement that gives later on the product, the antibody itself, occurs, the expression occurs on one of the chromosomes. The other pair of exactly the same sequence is not expressed. So the cell has to decide to choose one out of two. And it has a beautiful biological sense to it. So the body, the lymphocyte, these are the cells that make the antibodies, they need to make a very good antibody in order to help us to survive. Now, if we will make two antibodies per cell, we'll have a problem. Because if there will be two very good antibodies, then we are fine. But if, for instance, one will be a very good antibody and the other one will be a very bad antibody, still the cell will secrete both of them, and it can cause chaos in the immune system and cause diseases.
So is it because one antibody will find the other?
No, for instance, one antibody will be against a pathogen, which is a good antibody, and the other antibody will be directed against something in our body.
Oh, we'll turn on the body itself.
Exactly. So we'll have a very good anti-pathogen reaction, but we will have also an anti-self. It will attack our cells. So we need to make sure that for each lymphocyte, only one antibody is made. So if it's good, it will get what we call the signal for life, and it will live and will secrete antibodies. But if it makes a bad antibody, for instance, one that attacks our tissue, then this cell is going to be dead by a different process. So we eliminate all the cells that make bad antibodies, and we keep and cherish the one that make very good antibodies.
But there are some diseases which are related to bad antibodies, right, that attack ourselves. What is, that's not your area of research, is it?
No, we don't work on what you call autoimmune diseases. No, we don't work on particular autoimmune diseases, but we do have them in our thinking, because in some of them, this is the process that the bad lymphocytes were not selected against. So they kept in the reservoir of all the cells in the body, and they make bad antibodies. So these are the two main basic phenomena that create all our immune system: rearrangement and choosing one out of two.
And your research was really on the mechanics of how that happens, right?
Right.
Is it possible to explain that mechanics to a lay audience?
Yeah, Yeah, it is. So the two alleles, the two genes are identical, almost identical in sequence. So how do you choose one and not the other? So I'm sure you've heard from others about epigenetics.
Oh, yes.
This is what,
Well,
you know,
why don't you explain it to the audience?
the epigenetics is a level of regulation that is not connected to the sequence of the DNA itself, but it's rather the envelope that surrounds the DNA. And it has a very important role in affecting the regulation of gene expression. It can either help turn the genes on or turn the genes off.
The switch system.
Exactly, it's a switch system. So in a cell, we have two genes that are identical in sequence, but the epigenetics, the organization of one allele, is different than the other allele. So when the machinery, the molecular machinery, comes and looks where to do rearrangement, it sees one with a nicer and a better envelope, and it says, oh, this is a better substrate for me to go to. And it goes first to this allele, and it rearranges and then expresses.
It makes one the right hand and one versus the left hand, or vice versa. I mean, do you ever speculate on why we've built the system, or why nature has built a system like this, that has pairs and makes choices, why we're left-handed or right-handed, or, you know, on the macro level, but it happens on the micro level very particularly?
Yeah, in fact, you're absolutely right. It's a very good question, because I said that choosing one for rearrangement is unique to the lymphocytes, but expression of one of the two choices is not unique only to lymphocytes. All the olfactory system is like that. You just express one, although you have 1,000 others. And recently there is another study that says that 10% of our genes, it's an incredible amount, are expressed from one chromosome and not from the other. So the rationale can be different for the different tissues. But, so I just told what is the rationale for the immune system, and different rationale could be for other systems. But I do think that the building blocks of the mechanism, how it works, maybe share similarities among all systems, that you have choosing one out of two.
And why that is? I mean, it puzzles me as a philosopher.
Yeah, no, but for the immune system, it's easy to explain.
Because it is a functional purpose.
Right. For other loci in the genome, you have, not always we know what is the logic, but there are some important.
Well, we're going to take a break, but before we do, I want to signal what I'm going to talk to you about, because you then jump from your immunology work to stem cell work, and you don't initially see why the connection or how you did the connection. And so I'm going to explore that after the break.